My name is Swami Rajaraman. I'm an associate professor in material science and engineering, and also with an appointment in Nanoscience Technology Center, and secondary appointments in electrical engineering and biomedical sciences. My research, I'll start my clock here to make sure that I stay on time. My research, as from all these appointments that you can see, is extremely interdisciplinary. So my group is called the Nanobiosensors and Systems Group. And we are work at the intersection of biology, more in vitro biology, though we are getting more into the implantable and the wearable space. And my website, research website, is extremely up to date. So I'm going to rush through some examples of research in my group very quickly here. So if there's any questions beyond the Q&A here, please feel free to reach out to me or go to my website and connect with us. My email and phone number are also listed. In addition to research and teaching, I also have started last year serving as the Deputy Director of a NSF IUCRC called the MIST Center, Multifunctional Integrated System Technology. There's the website there listed as well. And this is a center where we welcome industrially funded projects, directed and collaborative research coming through the consortia for companies across the country and even across the globe. So if there's any ideas that fit the broad spectrum of integrated system technologies, please definitely come speak to me. We are looking to integrate new projects into that center as well. A very broad introduction to what I've done. I started at UCF in 2016. I got my Ph.D. at Georgia Tech. And then after that, I did a startup actually for a few years after that, which is also in this bioinstrumentation, biosensing space. But since coming to UCF, I've been very fortunate to get about 27 funded grants, total of almost 8 million in total with all collaborators and others as well. My group publishes around 7 to 10 papers in journals and peer-reviewed conferences every year. So we've published about 52. And given my industrial background, I'm also a big proponent of filing for patents. So we've had 4 awarded patents and several applications that are currently pending. 3 graduated PhD students, 3 graduated master's students, several postdocs. My group is typically 10 to 15 students at any time, including undergraduate students. We have a healthy dose of about 5 to 8 undergraduate students all the time. We actually have a current opening for a postdoc starting in a couple of months. So if you know somebody that's interested in some of this, please ask them to contact me as well. In terms of teaching, I've taught undergraduate classes in the fundamentals of materials science, as well as graduate classes in biointerfaces, which is the original class that I developed in Nano, and I teach laser materials processing as well in MSc. Some of the examples of the research in my group are shown here on the right-hand side, and I'm going to go through some of these details in the next couple of slides. I've also been building some credibility and hopefully some visibility for UCF in the MEMS and sensors community. I've served on the program committee for all leading MEMS conferences, and I'm going to chair a major MEMS meeting, co-chair a major MEMS meeting called the Hilton Head, a solid-state sensors, actuators, and microsystems workshop, the 40th anniversary of that meeting in 2024. And recently, I got elected to be on the editorial board of IEEE Journal of MEMS, which is the leading MEMS journal as well. So as I indicated, my group works at the intersection of micro-nano engineering and biomedical sciences. Of course, this area is extremely broad. So the 3 areas that we focus in within this are microphysiological systems. So as everybody knows, the FDA process for drug approvals is extremely slow. And if you look at the probability of success, if you start with 100 compounds, especially in the neural and cardiovascular space, after exhaustive animal trials and human trials and billions of dollars in spending, you succeed with 3 to 4% of 3 to 4 compounds. If you start with 100 compounds, that gets introduced to the public. So the field of microphysiological systems, where you recapitulate some of these signatures in a chip in order to accelerate these, and in some cases, maybe even eliminate this animal testing, has really exploded in the last 15 years. And one of the things my group has been able to do is to sensorify this microphysiological system, especially in the neural and cardiovascular spaces. And I'll show you some examples of that in the next slide. Since 2020, like many of us, we pivoted some of our research activities to researching pathogens as well. or accelerating, again, in this case, assays that are for viral detection. And we also do some bacterial detection that we've gotten some good funding from UCF Research Foundation to accelerate it to other kinds of things. And what we're doing here is to figure out multimodal sensing capabilities to sort of thread the needle between exquisite but expensive methodologies like polymerase chain reaction PCR testing, and sort of thread the needle between lateral flow assays, which are not that sensitive, but bring this point of need or point of use sort of systems closer to the user, but get better in sensitivity with things like ELISA and PCR. And of course, if you talk to my friends in the MEMS and sensors community, the answer is always silicon. Silicon has got an extensive, in terms of technological capabilities, very mature, a very mature tool set and the ability to do a lot of different things with silicon, which is wonderful. But my argument ever since I got to UCF, and even before in the startup, has always been, what about taking silicon and silicon technologies for where they shine, but integrate other novel methodologies and novel materials like polymers and different methodologies like lasers and printed circuit boards and things like that. And that's what we've been doing extensively in my group. And I call that hybrid or makerspace micro nano engineered sensors, but we are also starting to do more system level work, as I'll show in the next couple of slides. So the first one is the development of these three-dimensional microelectrodes. So microelectrodes are a grid of electrodes that are there in for spatially and temporally capturing some of these MPS extracellular signatures that I mentioned. So this is an ongoing activity ever since I came to UCF. I've had a very successful ongoing collaboration with Professor Michael Moore of Tulane, who's the chair of biomedical engineering there, Professor Randolph Ashton at the University of Wisconsin, who does a lot of stem cell differentiation, and some local collaborations with Dr. Zai, for instance, on some of the material technologies that we have developed together, and newer collaborations starting with Dr. Dubansky and Dr. Kim at Johns Hopkins. And what we have done, especially in the 3D electrode array space, is we have created 3D microelectrodes going from three different the simplest technologies, which is 3D printing. So ability to 3D print these 3D microelectrode structures. And you can see in this image, a 3D printed microelectrode array with a nerve on a chip on top of it. And you can see we did different viability and all of that to start with the system. And we ended up with some of these resin materials actually causing a toxicity to this nerve on a chip. But that did not, that still enabled us to create some really nice interfaces and also create some new materials, as is shown here in this 2022 paper, where we developed this polydopamine-mediated polystyrene as a 3D insulation for these 3D microelectrodes. But then we have pivoted to a glass technology, which we hope to further grow with our new collaboration for a cardiovascular system with Dr. Duck-Ho Kim at Johns Hopkins. But for our main collaboration for the neural nerve on a chip with Dr. Moore and Dr. Ashton, we have developed this polymer metal system, which is shown here with 5 sensing modalities that is inbuilt here, and this normal polydopamine-mediated silicon dioxide material, a material that usual MEMS and electronics communities do not like because of its porous nature and its poor dielectric properties. But what it has shown for us is the ability to actually anchor some of these gels that hold these microphysiological systems. And in this image, you see these microphysiological systems as dual spheroidal systems that is implicated in pain, actually. So it's a HEAL initiative opioid addiction project where you have this particular system which has these dorsal root ganglion synapsing on this dorsal horn being successfully grown on our, this is a control system, and being successfully grown on our MES for more than 6 months. So it's a very unique nerve chip, one of its kind in the world, where we can get electrical signatures like this, and we are starting to explore and grow these experimental set, and also grow the number of devices to be able to test a set of analgesics that will be not as addictive as some of the opioids. Of course, the outcomes, it's one of the first of its kind nerve chip in the world. We have gathered several months worth of neurological data, multiple NIH grants to support both the nerve chip and the cardiac chip, which are just ongoing, and some commercial efforts, for instance, with Axosim, which are interested in licensing some of these technologies. The second work that we started working mainly during COVID was with Professor Salaita at Emory University, and some overlapping efforts with Dr. Chernovsky and Dr. Santra here at UCF, was more on these pathogen detection systems. So for instance, Khaled Salaita has this extremely sophisticated assay called the RoloSense assay, where you have rolling microspheres on a surface. And when you introduce a virus, because of the surface engineering there with RNA and DNA and these molecular motors that are moving, when you introduce a virus, what they noticed was that sort of prevents this motion, and this motion completely stalls. And when you don't have a virus, and we started with COVID, that was our NIH-funded project, these motors keep rolling. So what we developed was the functional surfaces for it. We developed surfaces like microfluidic chips and this wireless imaging system that can track the motion of these motors, again, in a handheld system, which can be deployed, and along with a Wi-Fi enabled interface where we can actually track the motion of these systems and plot these pictures that the wireless imaging system captures into tracks that can then tell you whether whether there's virus in the system or not. So that's another effort that's ongoing. Currently, we also have a bacterial sensing system that we have been working for quite some time, where we integrate multimodal sensors like microelectrodes, interdigitated electrodes, and others for culturing and sensing different bacteria. And some of the work that we have done with Dr. Santra, for instance, also target bacteria and figure out their minimum inhibitory concentration. There's a recent paper that we published where we took a new spin on this disk diffusion test and developed this micro-stencil diffusion test for measurement of this minimum inhibitory concentration of bacteria. So this, again, we are building this into a biosystem that we can start developing with wireless electronics and other things in this space. And then the last category that I wanted to show was in this broad area, what I would call hybrid micro-nano-engineered biosystems. The reason I put biosystems in there is we are starting to go beyond biosensors and actually integrated system-level technologies along with sensors on the same chip. And this is something that we've been working on. I've been working on ever since I came here. One of the first things that we started doing was to develop microneedles, which are both passive diffusion-oriented and completely 3D printed and biodegradable. This is stuff that we have published with Professor Mandeep Sachdeva at Florida A&M, for instance, and also with Dr. Santra, where we demonstrated the world's first microneedle for injection of drugs into plants. This has never been done before, but people are starting to do this stuff now. Also, we did stuff with functionalizing microneedles, not just developing 3D printed microneedles that could completely biodegrade, but also functionalized microneedles, which could detect things like hydration on ex vivo models, like the one that we did here with Johns Hopkins on a pig model, and it was able to it was a metal microneedle that successfully showcased hydration sensing. But we've also done other stuff with on the system level with people like Dr. Tattard at UCF, Dr. Abdulwan at UCF as well, where we integrated different modalities. And these are things that we're starting to build on with new projects from a couple of local companies funded through this MIST consortium. But we're trying to take this data and make it easy to read and use. I feel like I'm running out of time, so I'm quickly go through my near-term and long-term goals. Near-term goals, of course, are to consolidate all the research teams, work on RM21 proposals to expand this nerve chip work that you're doing into the next step, and consolidate some of these R01 proposals that we have. The renewal of MIST for phase 3 is happening in 2024. So that is something that I'm looking to play a big role in as well. Graduate my current students, recruit new students. I'm in the process of doing all of that. And then I mentioned organization of this big MEMS meeting, so I hope to successfully organize that in 2024, and also play a role in the growth of the department. I'm chairing an infectious diseases faculty hiring committee in the department right now. Long-term plans are go towards more center-level grants for some of these ideas, spin out new ideas. I have a entrepreneurial background as well, so spin out some of these ideas into companies, and then increase my role in the MEMS community and create new classes, graduate classes as well as undergraduate classes. Some of the sponsors of my research, as you can see, they span from NSF, NIH, and also a lot of companies, local and beyond. These are the people that are responsible for all this work that I've showcased very quickly. Current group, alumni, and external collaborators. I'm always looking for new collaborations, especially within UCF. I've been able to do a lot of successful external collaborations, but would love to work with you on these ideas within UCF. And with that, an image of a collage of my group, I open it out to any questions that you may have.